Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our novel drug delivery systems, we are discussing on intrauterine and implantable drug delivery systems. In our third session, we are discussing on the osmotic drug delivery systems. So far, we discussed on these three topics, which is osmotic drug delivery system. We have seen the working principle and also we have seen the important points on the osmotic drug delivery systems. And then we are discussing, after that we have discussed on the structures of osmotic drug delivery system. Then we have seen the components of drug delivery systems. So, today we are going to discuss on the what are the ideal characteristics of osmotic drug delivery systems. Then we discuss advantage of osmotic drug delivery system compared with other type of implants, and then limitations, and last we will discuss on the types of osmotic drug delivery systems. So far we discussed on the components. Now we are going to discuss on the what are the ideal characteristics of osmotic drug delivery systems. So, the first character, that is drug related parameters, although we have discussed in the components the drug properties. Now here we discuss on very detailed on the drug parameters. So, as I mentioned in this, your drug need to follow moderate water solubility. That means it should have an optimum solubility in the water or in the GAT medium. So, the solubility between 50 to 300 mg for steady osmotic flow. Water insoluble drugs, we need to do the treatment or we need to do the processing. With that we will get a water solubility in the medium. So, yes, and drug should have the high potency. Because of here we need to use only low dose because of the space. For that we need to use high potency drug, and because of the limited core space in the osmotic devices. Stability in aqueous environment, the drug must remain chemically and physically stable after water enters. So, that we need to perform in the pre-formulation studies. So, then only we can select such drug molecules for the osmotic drug delivery systems. Now, as I mentioned, in the biological half life, the minimum is 1 to 6 hours half life should be there. Yes, there are the drugs which have short half life. Such drugs can be converted into sustained and prolonged release. With that we can enhance the biological half life in the blood. Consistent absorption site, preferably absorb in defined region of the GIT tracts. Example, upper small intestine. So, these parameters are very important for the drug properties, and definitely this your drug need to be compatible with all type of components which are used in the osmotic drug delivery systems. In ideal characteristic osmotic drug delivery system, we are going to see on the second topic, that is osmogen related parameters, that your system need to generate high osmotic pressure, and here these are the excipients we use: sodium chloride, mannitol, that will create a strong osmotic gradient. So, your system need to generate high osmotic pressure, and yes, the osmogens need to be compatible with your drug, should not interact or degrade the drug substance, that need to be physically and chemically should not affect your drug molecule. The third one is membrane related parameters, nothing but semi permeable membrane related parameters, and semi permeable nature, that should allow water in, but restrict drug and osmogens out. So, that is utmost important because water need to come through semi-permeable membrane. And biocompatibility, non-toxicity is a major requirement. Whatever material you are using like celluloacetate membrane, any other type of material that need to be safe for the human use. Controlled permeability should enable modulation of the drug release rate. Mechanical integrity is very important, that should withstand GI conditions, either it may be pH or it may be with some enzymes or it may be any chemical reaction which happens with the membrane that need to be withstand. So, without rupturing, otherwise if it is undergo any problem in the mechanical integrity, then the drug will release, it follows the dose dumping. So, that can be avoided. So, the fourth one is system design requirement. So, we need to provide a delivery orifice, so that with that precise and appropriate amount of the drug will released out from the orifice. So, resistant to GI conditions, that already we discussed in case of the semi-permeable membrane, should maintain performance regardless of food and the pH and motility. And yes, that should be scalable fabrication, system should be reproducible and suitable for the industrial scale manufacturing. And the fifth one is patient and product requirement. Once daily dosing is capability when you are oral systems, and reduced dose dumping risk that leads to the side effects, and also minimal side effect need to be come up as an ideal characteristics of the osmotic drug delivery systems. Now, the advantage of osmotic drug delivery systems. So, yes, your system need to produce control and predictable drug release, which provides the zero order drug release, which maintain the steady state concentration throughout the treatment, and that need to be follow delayed or pulse drug release, it depending on the formulation and type of formulation. So, independence from the body conditions. So, the drug release unaffected by the stomach pH, fluid movement in the GIT tract, and yes, the semi-permeable membrane coating system, which consistently will help in the internal condition that should not affect by any type of body conditions. The higher and adjustable release rate can be expected, that is one of the other advantage. Yes, faster drug release can possible compared with the traditional control release systems. Release rate can be programmed by designing various parameters like thickness and the orifice size. With that, we can also control the release rate of the osmotic drug delivery systems. Yes, and then yes, we can expect excellent in vitro in vivo correlation. We will predict by using the some mathematical and modeling systems. We will understand what is the correlation between in vitro response and in vivo release or actions. So, yes, the comparable release patterns and excellence in vitro in vivo correlation relationship is possible with the help of the osmotic drug delivery systems, less influenced by external factors like food, pH and the movement in the gut. So, with that we can expect IVIC with the formulations. Now, food has a minimal effect as we discussed, because of which comes in the as a carriers osmotic drug delivery system, that will not affect the release, the food will not affect the release of the formulation. So, drug remains protected inside the system until the drug is ready for the release. And easy manufacturing, that is large scalable production, that is one of the great advantage. Nowadays, there are so many products we have seen for the osmotic drug delivery systems. So, yes, we can possibly make the large scale production, we can shift R&D to pilot plant and then large scale, and for the commercial needs also possible. So, these are the some limitations. Yes, costly compared with the conventional or controlled drug delivery systems, because here we need to use various types of the polymers and semi-permeable membranes and other components, and we need to check lot of quality control parameters, and yes, we need to ensure the drug loading and the response of the osmogens, and that will help for the release. So, dose dumping is one of the major problem. Any changes happen, so that the entire amount of the drug will release at a time, like if undergo rupture or any leakage, any mechanical strength changes or variation happens, so that can be toxic to the patient. Possible hypersensitive reaction also possible in the patient because of the materials we are using, so that may undergo lot of hypersensitive reactions. Challenges in maintaining the product integrity and consistency, yes, that may vary from patient to patient, and also responding of that carriers within the human bodies. So, challenges in maintaining the product integrity and consistency also one of the major limitation in osmotic drug delivery system, because of this release will depends on the so many factors, size of the delivery hole or aperture or orifice, and also surface area of the system also very important, thickness and composition of the semi membrane, that will also affect the release. So, all these factors will leads to the limitation of the osmotic drug delivery systems. Now, we are going to see some time on the types of osmotic drug delivery systems. So, it can be classified into two major classes. So, these are the osmotic pumps, oral osmotic pumps, and the implantable pumps. Now, you can see these are the 5 types of the implantable pumps. You can see the Nelson model, and so Higuchi and Higuchi Thews and mini implants and ALZETs. So, in another side you can see the oral pumps or osmotic delivery systems pumps, that is single chamber, multi chamber and the modified systems. So, here we have few examples in the again single chamber, and also we have multi chamber system. Few of the we are going to discuss, and then rest of the information you can read it from these references. So, this reference is clearly explained on the various types of the osmotic drug delivery systems. So, we are going to discuss on the few examples one by one. So, the classification can be types can be classified into various systems. Also, we have discussed, that is single chamber osmotic, multi chamber, implantable. Again, elementary osmotic pump, control porosity, push pull osmotic pump, sandwich, and these are the OROS capsules. Okay. So, we are going to see this one also. Then ALZET pump and the DUROS pumps. So, in the elementary osmotic pumps, so here these are the single chamber osmotic pumps, nothing but here we have the only one chamber which contains the drug core, and also which is loaded with the osmogens. You do not see any other chamber, single chamber. So, yes, here the water enters through the semi-permeable membrane, dissolves the drug and osmogens inside, osmotic pressure builds within the tablet, and then pressure pushes the drug solution out through the orifice, which will provide the controlled and constant zero order drug release, and this is preferable for the highly water soluble drugs. Now, you can see here also in the picture representation, this is the orifice which can be made by the laser or by the mechanical process, and this is outside semi permeable membrane, and water is entering, and now the drug and osmogen will get solubilized, and then pressure will build. Now, that pressure will help to release the drug from the core to outside. So, this is elementary osmotic pump systems. This can be used for the highly water soluble drugs. Now, based on the Rose-Nelson pump, but simplified for tablet form. So, here tablet with a single layer core which contain drug and osmogens, which we already discussed, and that further coated with the semi permeable membrane. So, pre-filled orifice for the drug release also possible. So, these are the some marker product you can see which comes in the elementary osmotic pumps. Now, second one is controlled porosity osmotic pump. Now, you can see the title itself suggests that there is a formation of the pores which will help for the control release from the osmotic pumps. Now, you can see how it works. GI fluid enter through the membranes, pore formers dissolves and leaches out. So, this point important after entering the water through GAT fluid, and then it will enter into the inside of the capsule or inside of the osmotic pump, then it will form the pores, that is called as micropores, which are there in the coating membrane or semi permeable membrane. So, now you can see here, so this is the carrier, and you can see once water is entered, what happen, you can see there is a formation of micropores. So, this system will not contain any orifice. Now, you can check everywhere, you will not see any orifice anywhere throughout the system. So, these micropores will work as an orifice. So, that will release the drug from the inside to outside. So, water enters the core, drug dissolved, drug is released through the pores. So, there is no single orifice, the release occurs from the entire surface, not just one point. So, with this you can expect high amount of drug release from the system. So, there is no drilled orifice required, pores forms automatically, water leaches out pore forming agent like example of sodium chloride, urea, sorbitol, PEG can work as a pore forming agents. Drug core contains drug and osmogen, this is coated with the semi permeable membrane which contains the preformers. Yes, definitely which has a high advantage compared with the elementary osmotic pumps. Now, in the multi chamber, now you can see in the multi chamber you will see two different chambers here. So, here is the one and here is the 2, right. So, it contains the two different chambers, right. So, this can be separated, you can see here the swellable polymers and osmogens, here is the drug reservoir. Now, this is a delivery orifice. So, once it is given into the body, so water enters to both layers through the semi permeable membrane, water enters from this and also this. Okay. So, after that the drug layer forms a saturated drug solution, the push layer. So, now you can see push pull. So, this once the water is entered into the osmogenic area, so this swellable polymer also swells, that leads to create the pressure or which will push the drug chamber. So, with that the drug will release. So, push layer swells and expands due to water uptake. The swelling pushes the drug solution out through the orifice which will provide the control and constant drug release. Now, you can see, so this will swells and also which will expand, then due to the swelling, so it will release the drug. So, one is the push layer, another one is the drug layer. So, push layer will help for the pushing and delivery of the molecules. So, yes, which can come in the like a bilayer tablet coats, the drug layer which contain the drug and osmogens, and the push layer contains the swellable polymer and further coated with the semi permeable membrane, has one delivery orifice on the drug side. So, you can see there are the products which comes in the type of push pull osmotic pumps, that is glucotrol made by the Pfizer, and the Cardura XL, that is doxazocine also from the Pfizer. So, both will help in the prolonged release. The second one is in the multi-chamber osmotic pumps, the sandwich osmotic tablets. Now, here you can see, you will see the two layers, right. You can see here, this is the drug layer in the middle. In the middle we will see the push layer, and the outside is drug layer. So, here we can consider as a sandwich system. So, water enters the tablet from both sides like here and also here. From these two sides, so water will enter into the push layers and then it undergoes swelling upon the hydration. The swelling creates a pressure on both drug layers. So, once the water is entered, this push layer will help to push the drugs from the inside to outside. So, this core are in the between in the sandwich. So, the push layer is placed in the two drug layers. So, that is why it is called as a sandwich osmotic tablets. So, once the water is entered, then from two sides it will swells and from the up and bottom are two sides. So, the drug will release. Now, you can see in the pictorial representation. So, this is a before operation or before treatment, after this is after administration. So, here you can see the water is entering. Now, it will help in the pushing of the drug molecules from the core systems. So, the drug will release from the both side. So, yes, in this case you can expect high amount of drug release as well as control system throughout the treatment. So, here we have two orifices in these systems. So, key features, it contains the 3 layers, one central layer of push layer and the both sides you will see the drug layer, 2 delivery orifices, one central push layer, 2 drug layers and also 2 delivery orifices. Total 5 systems are there, which is in the sandwich system. Now, osmotically controlled release oral drug delivery systems. So, here these are considered as a capsules. Now, you can see in the capsule format, these are the before operation and the during the operation. So, these again further classified into OROS, CT, that is nothing but colon targeted osmotic systems. They will respond in the colon specific delivery, that is enteric type of formulations. These have multiple push pull osmotic units in a single capsule. So, this coated with the enteric layer. So, that will bypass the degradation and some other problems in the stomach pH and they will release only in the colon. Ensure targeted and delayed drug release throughout the system. Now, you can see, so this is the outside rate controlling membrane, the osmotic layer and the inner layer, and this is a soft gelatin, and inside we can provide the liquid drug formulation as we prepare like a soft gelatin capsules. So, these are considered in the category of the soft gelatin capsules, but which are osmotically controlled release oral delivery systems. So, once the water is entered, then it will swells and that gelatin will help, the soft gelatin capsule of the osmotic system will help for the release of the drug. Now, you can see the expansion of the osmotic push layer in the capsules. The second one is OROS system, which are designed for liquid drugs and other macromolecules for the poor soluble and instability molecules. Now, the another system is ALZET pump. So, these are the another systems. Now, these contain miniature pump with drug reservoir and osmotic sleeves and also the membrane. And yes, with this you can expect continuous drug delivery compared with other type of osmotic drug delivery system. You can see here other systems can be used for 1 day, 2 days, but these ALZET pumps help for the 1 to 6 weeks. So, these many people are working in this research application of with the ALZET pumps, and also accurate consistent dosing also possible. It will also reduce the animal handling and stress conditions, because we can use it for the even animal research purpose also. and yes, this can be good for the short half life drugs and can target the specific tissues also. You can see the label of the ALZET mini-osmotic pumps. You can see here the 42 days can be utilized for such systems. Now, you can see here which contain the semi-permeable membrane, osmotic layer, and you can provide your solution into this. These are the impermeable reservoir, there are the flow modulators, and then you will see the release through exit port. DUROS systems, which are also there are the products available in the market. You can see here via DUR, which contains the DUROS system, which will help for the prostate cancer. We know the cancer is the required the chemotherapeutic treatment for the long time, because which comes under the chronic treatment. So, which contain titanium encased rod shaped implant. Now, you can see in the picture. So, water enters, osmogen agent swells, then piston pushes the drug out. Now, you can see here. So, this is the semi permeable membrane, right. Water will enter here, and this is a osmotic engine and then piston. So, the pressure builds, and this piston will help in the release of the drug from the orifice, and it works on the diffusion moderator. The entire system is reservoir, and here it will contain the drug. So, yes, it can use for the 1 years for the treatment of the various diseases. Advantage, delivers a viscous and the concentrated formulations also possible. High precision control is possible, which will protect from the all type of degradation in the body. And yes, you no need to take daily doses. You can use the DUROS system, which will help for the 1 year. With that, we can improve the patient safety and as well as compliance. So, there are other osmotic delivery systems also available in the market. So, which are used for the treatment of various diseases, like as we have seen in the cancer, also diabetic, in the neurological diseases also we can be used. Even in case of infectious disease also we can use various types of the osmotic delivery systems. So, with this we are going to stop today's lecture. So far we discussed on the implantable drug delivery systems, and also we have discussed on the osmotic drug delivery systems. In our upcoming classes, we are going to discuss on the intrauterine drug delivery system and other some type of the drug delivery systems. Thank you so much.